have no clue which direction evolution is happening, right? So that is the most important reason why trees have to be rooted. So you can have an unrooted tree, which looks more like a network actually than a tree, right? But you cannot make out which direction evolution is happening and so on and so forth. That's one of the most important reasons we have to root a tree, right? Okay, I'll move on to the next slide. So this again shows you like the three different structures. Like on the top, you have an unrooted phenogram where A and B are sisters and C is sister to A and B. The little oval here basically tells you that this is the longest branch in the phylogeny. And if you want to do a certain kind of rooting, you have to use the longest branch. We'll get to that. And then you representation of that unrooted tree as a slant phenogram. And again, right here, you can see the direction in which evolution starts to happen, right? And similarly, you have a rectangular phenogram that represents the same topology. All right, moving on. Okay, here are some terms that you need to familiarize yourself with in order to be able to converse about trees. So for example, when you're talking about a phylogeny to someone else, you need to be able to freely use and understand some of these terms that I'm going to talk about, right? So there are two types. So basically, there are nodes and branches in any tree, okay? A node is basically a point at which there is diversification in a tree, right? You see this that I'm pointing to, that's a node. This is a node 2, and this is a node 2, which is the root node, okay? These nodes are called internal nodes. Similarly, the nodes at the, at the leaves of the tree, they're called terminal nodes or external nodes, yeah? But these also are nodes. So you have two kinds of nodes. You have the internal nodes, and then you have external nodes, right? Similarly, you have internal and external branches. So an internal branch basically connects you to an internal node, whereas an external branch leads you to an external node or a terminal node. Some people call the terminal node a leaf too, but we would just stick with terminal node, right? Is everyone clear with the terminologies here? And the root over here represents the oldest ancestor of this group of taxa, right? And every internal node represents an ancestor at a certain point in time, right? So for instance, for the capped langur and the golden langur, we would hypothesize that their most recent common ancestor is at this node, because from this node there was diversification, and one lineage led to the golden langur population, the other one led to the capped langur population, right? So yeah, so these are some terms that you will need to start using and using commonly when you want to talk about phylogenies. Similarly, the shape of the tree and the branching order, right, or the relationships of a tree, the relationships that a tree represents, is called the topology of a tree, right? So like we saw in the previous slides, that you might have the same set of taxa, but the topologies could be completely different. So you have to keep your eyes very open when you look at two different trees to actually find out if the topologies are the same or not. With 4 taxa, 3 taxa, 5 taxa, it's fairly straightforward. But when you're looking at like 10 taxa plus, 10 taxa or more, it can be a little tricky to be able to, tell if the tree topologies are the same or not, right? Okay. Any questions so far? Cool. I'm going to move on to the next slide. Rooted versus unrooted trees. We briefly saw what unrooted trees were in a couple of slides ago. So the root, like I said, is the point in time that represents the earliest time in the evolutionary history of these sequences, or the oldest perceivable ancestor for this group of taxa. An unrooted tree, as the name suggests, does not have the root, and an unrooted tree looks something like this, right? Now, from the tree on the left, I'm sure you can't make out much, right? It's very hard to decipher what's going on. You can make out a few things, but you can't make out broad relationships unless you look really hard. And obviously, you can't make out which direction evolution is happening in. However, on the tree on the right, it's all very clear, right? You know exactly what's going on. You know that some lineages have accrued a lot of evolutionary change, as opposed to some other lineages over here that have that are not very divergent from each other, right? So this is why all trees have to be rooted. And on the bottom of the tree, you see what's called a scale. Yeah. So the scale represents, it's like a scale in a map, right? So I would say that this length represents 0.02 evolutionary changes, right? And so you can actually look at the length of a branch and kind of approximately figure out how many evolutionary changes have occurred. Yeah. Okay, let me move on to the next slide. So most phylogenetic methods produce unrooted trees. Like we briefly saw UPGMA yesterday, which is probably the only method that produces a rooted tree, right? Every other method that you will use during these sessions, especially, every other method will give you an unrooted tree, and it is our job to basically root the tree, right? And we're going to talk about how to root a tree in the forthcoming slides. So this chart also talks about how none of these methods have a way of figuring out the direction of changes. They only base their, the differences between sequences or even morphological data based on the number of changes, right? So we'll look at how to root a tree in the next couple of slides. Okay, why root trees? So including the root improves the estimate of the phylogeny, and it lends the phylogeny a certain degree of stability. Okay, so if you only have, okay, let me not get into that right now. Maybe on the next slide. And obviously, the root gives you direction of evolution, and it's useful, especially when you're trying to construct character evolution of organisms and things like that. The two methods used to root a tree, the first one is called the outgroup method, yeah, and the second one is called the midpoint rooting method. Okay, so the outgroup method is actually the most preferred method, and everyone uses the outgroup method today. So let's assume you have a bunch of taxa. Let's say you're working on some gecko genus, okay, and you've assembled sequences of all the species in that genus, and you want to build a phylogeny of it. So in a case like that, you would, for the outgroup, you would typically pick a gecko from another genus that's most closely related to the genus that you're studying, right? And the genus you're studying and the genus for which the phylogeny you want to build is called your in-group, right? So, for example, if I want to build a phylogeny of hemidactylus geckos, I would probably use a Certodactylus gecko as an outgroup, or a bunch of Certodactylus geckos as my outgroup, right? Because I know from prior studies and experience that Certodactylus are the most closely related geckos to Hemidactylus. So similarly, you can find the right outgroups for your model organism, right? For example, if you want to build a phylogeny of all apes, like what we saw yesterday, which includes humans cladistically, you would probably use old world monkeys as the outgroup, right? Because they are most closely related to apes, right? The midpoint rooting method, basically you look at an unrooted tree and you figure out which the longest branch is, right? And you pick the longest branch and you find the midpoint of that longest branch and root the tree at the midpoint of the longest branch. So that's what midpoint.
Chaitanya, can you hear me?
Yes.
We have a couple of figures of outgroup and midpoint routing, no?
Yeah.
You might as well move to those slides.
Sure. Right. And midpoint routing also assumes molecular clock, right? Which is a big problem. I think, like Praveen was saying yesterday, we cannot assume that all lineages are evolving at the same rate, and that is against the tenets of evolution itself, right? So I moved on to the next slide, where we talk about how we choose an outgroup. I think I already spoke about this. So if we are trying to build a phylogeny of apes, and that is your in-group, you would choose old world monkeys as your outgroup, right? So if you're, and then you have, choosing the right outgroup lends a lot of stability to your phylogeny. Okay. So think of it as looking at a photograph. For example, if you look at a photograph of an insect, right, and someone has taken a nice macro picture of an insect, you have no idea of size, right? You have no idea how large or small that insect is, unless someone places a scale there, or someone places their hand or a coin or something like that, just to give you a perspective of size. So you can kind of visualize an outgroup that way. It kind of tells you the extent of change that has happened in your in-group. It kind of lends you that perspective. Okay, and it also gives stability to your phylogeny, and it roots the tree, which means that we have now a direction to look at, in terms of evolution. It's very important to choose the right outgroup. I have to spend a couple of minutes on this. For example, when your in-group is apes, you can't choose cows as your outgroup, for example, right? Now that's because that's evolutionarily very far away from apes. Or even worse, you can't choose crocodiles as your outgroup. Similarly, if your in-groups is apes, you can't use another ape as an outgroup, because that obviously is part of your in-group itself, right? So let's say I'm just building a human, chimp, gorilla, orangutan, let's say I'm building a human, gorilla, orang, gibbon phylogeny. I can't use chimps as my outgroup, right? Because chimps are part of the in-group. So you have to be very careful and judicious while choosing your outgroup, and traditional taxonomy really helps. So consult a taxonomist to find out what the closest group to your in-group is, and choose an outgroup from there. Okay. So now we'll talk a little about midpoint routing, which is not very commonly used. So in an unrooted tree where you already have an outgroup, let's say, the midpoint would fall on the branch that connects the in-group to the outgroup. Very simple, right? You already have an outgroup here, langurs and baboons, and they are outgroup to the group of apes, and this is the branch that connects the two. So your midpoint would be somewhere here, and you can just use this midpoint to root your phylogeny, right? And once you root it, it would look something like this, very similar to the other type of routing. But the problem, like we spoke about with midpoint routing, is it assumes that every lineage is evolving at the same rate. Okay. Okay.
Hi, Chaitanya, can I ask you a question?
Sure.
Jayaditya here. That outgroup thing that we were discussing here, just that slide where we had this outgroups.
Yeah.
In that outgroup, you took like two outgroups, like baboon and langurs.
Yeah.
My question is, like, I see lots of paper where they are using a single outgroup or a group of outgroups, and sometime maybe of same genus or sometime maybe of different genus in the outgroup. So is it? Can you just deal with this a bit?
So at times you're not really clear what the closest sisters of your in-group are, right? So you could use multiple genera as your outgroup, right? And sometimes you would want to use a sister lineage as an outgroup, or an older sister to that whole clade. So typically, when you're not really clear about evolutionary relationships in your group, you can probably use multiple genera as outgroup, right? So you don't miss out on the right phylogeny. Does that answer your question?
Yeah, I think it answered for as of now, but as we go on, maybe I can ask some more.
Sure.
Thank you.
So I think one thing that Jaditya asked is he's also seen papers where they use the same genus as an outgroup. That was part of your question, right, Jaditya?
Yes, true.
Okay,
And
so
also conversely, there are certain multiple outgroups. Sometimes it's the one genus, many species of the same genus they put as outgroup. Sometimes there are outgroups that belong to different genus. There are two situations. That's what I was pointing out.
right. So it really depends on the question you're asking and the kind of phylogeny you're building, right? Let us say for example, because I know geckos, I keep reverting to gecko examples. Sorry about that. But let's say you're building a phylogeny of Hemidactylus frenatus. Okay, Hemidactylus frenatus is a really tiny, nondescript gecko found all over India. Now you're building a phylogeny of Hemidactylus frenatus from across its range in India, right? In such a case, you would choose an outgroup that is related to Hemidactylus frenatus, but it is not Hemidactylus frenatus. Like, for example, Hemidactylus leschenolti, right? So here's a case where your in-group and the out-group are from the same genus, primarily because the question you're asking is different, which kind of impels you to build a phylogeny of Hemidactylus frenatus, right? So your out-group will be something like Hemidactylus leschenolti. Right. However, if you're building a phylogeny of all Hemidactylus in India or all Hemidactylus across the world, your outgroup should definitely be a different genus, and it can't be Hemidactylus again, right? So it really depends on what kind of questions you're asking and what you're building the phylogeny for. That really determines the outgroup. Does that answer your question?
Yes, thank you.
Okay. So let's say I try to build a phylogeny of all humans in the world, hypothetically speaking, right? My outgroup would be chimps. I mean, I know they belong to different groups or different genera, but that's how I get you. I guess you get the idea, right? Cool.
Hello, Chaitanya, it's Shubham here.
Yeah, sure.
Yeah, so if you have an option for example, when you are doing phylogeny for Hemidactylus, you have an
Yeah.
option just for the sake that you have Cerdactylus also and Dravidogecko also. So
Yeah.
so if they are just as equal, there is nothing much difference in that. How would you choose any one of them? That this should be my outgroup.
So, like you said, Dravidogeckos are sisters to Hemidactylus, right? However, there is a certain, some gecko people in the world believe that Dravidogecko are actually Hemidactylus itself. They though they have a different genus name, they're part of the same hemidactylous radiation, right? So it's basically to dispel such people who have such questions, right? So I would rather include the immediate sister, which is the Ravidogecko, and the slightly older sister, which is Certodactylus, right? Does that answer your question?
Yeah, it does. So this means that we are not taking the just sister, but we'll go a bit more further.
Yeah, it's safer to just do that, because
Okay.
it's not going to affect your results.
Okay. Okay. Yeah, got it. Thank you so much.
Yeah, I think we spoke about midpoint routing enough. So this slide, I hope all of you can see this. This slide talks about the difference between morphological and molecular data. So one of the big problems with morphological data is how much data can you actually record, right? If you look at most modern morphology papers, they probably have 20 characters at the max, if you notice, right? And let's say there's some really diligent taxonomist who wants to look at anatomy, look at skeletal structure, the works. Then you'll probably get maybe 20 more characters at the max. So in effect, you have, let's say, 40. If you do a really thorough job, you probably have 50 morphological characters. Now, are these 50 morphological characters enough to build a robust phylogeny, right? The answer is no, because these are very few, actually. If you look at most modern phylogenetic algorithms, they expect way more than 50 characters. Similarly, how do you code these characters, right? If you remember, Praveen yesterday spoke about synapomorphies, convergent characters, unique characters, and so on, right? So unless you're a taxonomist who knows this group in and out, you will not know which of your 50 characters are synapomorphies, right? You will not know which of your 50 characters have evolved because of convergence, and which characters are unique, and so on. So that also has to be taken into consideration when you're building phylogenies using morphological data, right? One of the other problems with morphological data, I think yesterday someone was asking this question about cryptic species, right? We are finding more and more that a lot of the species in India, a lot of the groups in India are in fact cryptic. So when I say cryptic, I mean that there is negligible morphological difference when you just look at them superficially. So if you see photographs of two different species, you'll not be able to tell which is which and who is who, right? So lack of variation is another big problem with morphology. The only advantage is morphology is usually inexpensive because nobody bothers to pay taxonomists, and taxonomists would generally come and work for food, right? So it's usually inexpensive to hire a taxonomist to do that kind of work. The advantages of molecular data is that you just have a large number of characters. Every gene that you sequence has, some of the genes have excess of 1,000 characters, just that one gene, right? And it's very easy to code molecular data because there are only, 4 possible nucleotides at any site, right? And there is a lot of variation in molecular data because random mutations. We are finding out that we're finding out more and more that random mutations occur more often than it was previously thought. And so therefore there is a lot of variation with molecular data that you can use to discern divergence between species, right? The other big advantage of molecular data is you can build a phylogeny of absolutely anything. You can build phylogenies of geckos, of cetaceans, of bacteria, of viruses, of anything, right? And here's the cool part: you can actually put all these groups into one phylogeny, right? As long as you're using the same genes to build the phylogeny, as long as you've sequenced the same genes, right? So you can put all these groups in the same phylogeny and build what's called a tree of life. So that's the cool part with molecular data, right? And the techniques for sequencing, the techniques for building these phylogenies are pretty much the same across taxa. So it's completely agnostic to your model organism. Molecular data is also less prone to errors, and it's also less prone to individual biases. For example, if let's say there is a group of lizards and there are two taxonomists looking at the same group, right? One guy would say, oh, that crest on the head is a very important morphological character. The other guy would probably say, no, that crest is a very plastic variable character in this group. I don't think it's an important synapomorphy in this group, right? So you can have a bias between two taxonomists, both of them dealing with the same group. However, with molecular data, you can't have such biases. Let's say at site 123, in a certain sequence, there is a C. It is a C. Another phylogeneticist cannot interpret that site differently, right? So it is completely bias-free that way. The disadvantage of molecular data is it's very expensive, especially when you want to do when you want to sequence full genomes and things like that. So evidently, as you see, there is a great advantage in using molecular data in building phylogenies, as opposed to using morphological data. So all modern day phylogenies, whatever you pick like 100 papers that have come out in the last few months, everyone would have built phylogenies based on molecular data. I don't think anyone ever builds phylogenies based on morphological data. So what does that really mean? What it means is we should all shun morphology, right? Forget morphology and let's only do molecular systematics. Does everyone agree with that? Let's just get rid of taxonomy because it's redundant and useless, and it's too prone to bias and interpretation. So yeah, so there are, you obviously cannot shun taxonomy. The bottom line is taxonomy is still the bedrock of biology, right? If you're dealing with a group of organisms, you have
to Chaitanya, many people disagree with that statement. By the way, I just saw the messages.
So they want to get rid of taxonomy, or they want to keep taxonomy?
They want to keep taxonomy.
Okay, good.
At least that's the way. Maybe one of you guys can speak up.
Chaitanya, Jayaditya here.
Yeah, Jay.
So what I believe is even with molecular thing, we are doing taxonomy only. So this is molecular taxonomy, that is morphological. The base or the support is in morphology. But the most important problem with molecular data, that as a morphological taxonomy I like. So I believe that if I don't see the animal, the difference is I cannot randomly keep on picking all the geckos out of the wild. So I have to see certain things of interest, which is in morphology, that interest me to pick it up and do the sequence. So so I believe that the thing that I see is very important, and then comes the what I decide to do morpho. And you also yourself says that if I do for a species level thing, molecular is not that important, but if I do want to know about what is the relation to the rest of the taxa in the genus or in the family, then molecular. But if I have to just separate one species for another, I think still today the morphological taxonomy holds out.
Yeah, you're absolutely right. I can't disagree with that. But however, how do you deal with cryptic species, right? Species where you can't tell the difference at all. So in such cases, molecular data gives you another line of evidence to be able to delimit cryptic species. But remember that molecular data just gives you another single line of evidence, just like morphology does, just like geography does or ecology does, right? I think it's important here to, mention that morphology and taxonomy are not synonymous words, Okay. Morphology is the study of the external shape, size, characters of an organism, okay? Or internal internal anatomy, osteology, and things like that. Taxonomy, on the other hand, is the science of classifying and naming animals, right? Now, taxonomy could be just based on phylogenetic classification. That is still taxonomy, right? So, taxonomy is the science that classifies and gives organisms names, whereas morphology is, the study of size, shape, characters, and so on, right? I hope that is clear. Because I've seen a lot of people using these two terms synonymously, and they're not really the same, right?
But in case also natural history is also an important role to play to understanding all these factors.
Absolutely. Absolutely. So natural history, like you mean ecology and behavior and things like that, right?
Yeah, Chaitanya, I wanted to add that molecular data, if you want to find out evolutionary relationship between species, molecular data is the way to go. Because of the issues with morphological data, you don't get enough information to build a robust phylogeny. However, you still need, so-called traditional taxonomy, morphology-based traditional taxonomy to identify your tips, right? So for that, anyway, you can't run away from taxonomy. Morphology-based taxonomy. However, if you want to build the evolutionary relationship, then molecular data is the way to go, because you get a lot of information, there's a lot of variation. And usually the problem of convergence that you see with morphology is not a huge problem with molecular data, right? So basically, I think both have to go hand in hand.
Yeah, thanks for that, Praveen.
Yeah, and also I mean, the most number of morphological characters that, at least I know that the most number of characters that any paper has used is 300. And in order to get 300 characters, you'll have to shell out an insane amount of money, because you're not just talking about the external morphology, you're talking about some other characters as well. But on the other hand, molecular-based taxonomy, it might not need as much money.
Yeah. Yeah. So, any other questions, guys, on this slide? So I just missed talking about this. If you look at the figure on your right, it basically talks about 3 organisms, 1, 2, and 3. If you build a phylogeny based on morphological characters and say that color is a very important character for this group, right? So I will group the 2 red-colored organisms together and say they are sisters, and the blue-colored organism is sister to that. So you get a phylogeny that looks like this, right? However, if you decide that shape is more important than color, you'll get a phylogeny that looks like this, right? So this is again a big problem with morphological phylogeny is built with morphological data. I think we briefly alluded to this. If you don't know how to classify your morphological characters into synapomorphies, convergent characters, unique characters, and so on, building that phylogeny is going to be hell, right? You're going to be you'll end up with the wrong tree, and you'll start making grand assumptions based on the wrong tree, right? If no one has further questions on the slide, I think we can move on to today's section or today's slides. Okay, so this is the bottom line or the takeaway from the previous lecture. The phylogeny is an evolutionary tree that shows how different species are related to each other, right? A molecular phylogeny is a phylogeny that's built using molecular data. And what is molecular data? It's basically DNA sequences. And this is how DNA sequences look like. And in today's session, we'll look at 2 distance-based approaches. They're very simple approaches. They're called UPGMA and neighbor joining. We did a basic UPGMA thing yesterday with those green colored frogs with fancy characters and all. Today we'll do the same UPGMA exercise, but with using molecular data or DNA sequence data, right? So we'll move on to today's sessions. Okay, in today's session, we're going to cover methods of tree building using distance approaches, right? There are 4 or even more methods that use distance-based approaches, but we will only cover UPGMA and neighbor joining today, and we will kind of ditch the rest because nobody really uses them anymore. It's not like people use UPGMA and neighbor joining either. I've actually seen a few papers that use neighbor joining even today, but it's not a very accepted approach. The reason we're doing UPGMA and NJ is to show the difference between the two, the kind of trees that both distance methods generate, and we'll jump right into it. Okay. So this is a flowchart that shows you, the typical tasks of getting from starting to collect your samples to building your tree, right? The picture on the left is Praveen, I think, during his PhD days. Okay, I know what you're thinking. The person on the left is Praveen. So you start with sample collection, like what Praveen is doing in the field right here, and he's sitting right next to his model organism. So sample could be like, hair, blood, tissue, scat samples in case the animal is protected and so on and so forth. You'd have to make do with the animal's scat. And once the sample is collected, you'd have to bring it back to the lab and use a pretty rigorous process to be able to extract DNA from those samples. And then you go through what is called PCR amplification of the markers that you're interested in. By markers, we really mean genes here. So based on the questions you're asking, you would have chosen nuclear markers and or mitochondrial markers. And so you try and amplify those markers in your during your PCR amplification process. And once you have amplified, you basically sequence the markers that have been amplified, right? And you get what are called DNA sequences after that. So a DNA sequence basically is a string comprising of nucleotides, and like all of you know, the 4 nucleotides are A, T, G, and C. Yeah. And once you get your sequences from the marker of your choice, you have to align those sequences, and we'll talk about alignment and all in the practicals. This is a very important step, and it's basically the most fundamental step before you can move on with your data analysis. Right? So post-sequence alignment, you do data analysis where you gauge molecular evolution and you look at a molecular phylogeny of the samples that you have collected from the field, right? And once you've done your data analysis, you have your phylogeny, you build your trees, and you write your paper and you publish, right? What this flowchart does not tell you is at the end of this process, you get a Nobel Prize. Okay? Yeah, a Nobel Prize for peace, maybe, but nothing more than that, right? Okay. Anyone has any questions in this with this flowchart?
Hello.
Yeah.
Whether peel skin can be taken as a sample for DNA extraction?
Sorry?
Peeled skin.
Peeled skin. Yes, I think it can. Yeah.
Thank you.
Okay. This is what a sequence alignment looks like. A typical sequence alignment.
Hello.
Yeah.
Hello.
Yeah, I can hear you.
So, what's amplification occurs? I mean,
It's not properly audible. Maybe if you can type out your question, I can read it out.
okay. If something so we could.
It's not audible. It's not audible. So just type out your question and I can read it out to Chaitanya.
Well, let's move on. I clarified his question. Basically, the question I think was, PCR amplification of markers. I just told him that, we are not going to cover the molecular methods in this workshop. We are assuming that we have the sequence data and then we'll just move from there, how we are going to analyze the sequence data. We are not going to talk about how the sequences are generated.
Yeah.
Okay, Chaitanya, I have a question.
Yeah. Go ahead.
Like, what kind of DNA marker is suitable for building a tree, phylogenetic tree?
We'll get
Like, suppose
we'll get to that later in the course, especially on the last day, the frequently asked questions, right?
oh, okay.
We talk about what kind of questions, require what kind of marker and so on, right?
Yeah, fine.
Oh, cool. Okay, so this is a typical alignment of langurs. All you need to figure out from the slide is on your left are the terminal nodes or the taxa, and these are so against each taxa you'll see a sequence of nucleotides, and this is from a mitochondrial gene called cytochrome B, right? So when you align your data, this is how it pretty much looks like. Okay, just going to move on. Okay, so let's try and build a simple UPGMA tree based on sequence data. On your left, you have 4 taxa: humans, chimp, gorilla, and orangutan. And in the columns, you have 9 characters or 9 sites from DNA data, right? From a DNA sequence. So each of these numbers represent a single site, and these are the sequence that correspond to the taxa that we're talking about. Now, just like what we did with the morphological UPGMA method, we calculate distances between pairs of organisms here, right? Or pairs of taxa that you've chosen. So for instance, if you look at the snippet of the sequence that we have for human and chimps, the number of differences between human and chimp is 2. Okay. Similarly, the number of differences between humans and gorilla is 4, and gorilla and chimp is 3, and so on and so forth. So it's that the same dissimilarity matrix that you built yesterday with morphological data. Okay, going to move on. And just as you did with morphological data, you pick the taxa pair that are closest to each other from the dissimilarity matrix. And from this matrix, you'll see that humans and chimps are closest. So you pick them, and they are the difference between human and chimps is 2. So you basically divide 2 by 2 because there are 2 organisms, and the branch lengths that connect humans and chimps are of 1 difference each, right? Simple enough, right? Now, you've now bunched human and chimps together into a node called HC. That's become a single node now. You need to now calculate the distance between HC and the remaining organisms, which is basically HC and gorilla and HC and orangutan, right? So the difference between the distance between HC and gorilla is the actual distance between the human and gorilla plus chimp and gorilla divided by 2. Because we are taking 2 distances here, we divide that by 2, which is basically 3.5. So the calculated distance between human chimp, which is now 1 node, and gorilla is 3.5, right? Similarly, you get the distance between human chimp and orangutan, and you calculate that, and that is 6.5. The distance between gorilla and orang remains 5, so you leave that as such because we have not yet dealt with those two. Now, in this matrix, which is the smallest, which pair gives you the smallest distance, right? So evidently it's human chimp and gorilla. So that's the distance of 3.5. So now you have to like build a branch that connects gorillas with humans and chimps, right? So you take 3.5, divide that by 2, you get 1.75, and you build this branch that you see here, right? While this branch is of length 1.75 or distance 1.75, this branch is of distance 0.75 because 1 is already accounted for in this human chimp clade. Is that clear? So in the end, from the root, right, every terminal node is of distance 1.75 from the root, right? That is the idea over here. Now, from your dissimilarity matrix, you pick, you've bunched up humans, chimps, and gorilla into one clade or one node, and then you have to find the distance between that node and orangs. And you just do that by calculating the actual distance between human and orang, chimp and orang, and gorilla and orang. Divide that by 3 because there are really 3 distances here. So we divide that by 3, and we get a total distance of 6. Right now, we add the branch that connects orangutan to the clade containing chimp, human, and gorilla. Right, and we know that the distance is totally 6. So we divide the 2 branches into 3 and 3. Right. So now when you notice, the distance between the root and every terminal node is 3. Right. It's 3 along orang, and over here it's 1.25 plus 0.75 plus 1. And similarly over here is 1.25 plus 1.75. Right. So finally you get an observed distance matrix like what you see in the bottom over here, right? So this is what we've calculated for humans, chimps, gorillas, and orangutans using UPGMA, right? So the reason this method is called UPGMA is because we pick taxa in pairs. So that's where the paired group method comes from. And the distance between the taxa that we pick, for example, human and chimp, is 2, and we just divide that by 2 to be able to arrive at branch lengths here, right? So basically what we're doing is we're picking the mean branch length and assigning it to each branch that goes on each lineage. So that's where the arithmetic mean component of UPGMA comes into play, right? I'll move on to the next slide. So in this slide, you can see that the observed distance, which is basically distances we acquired from DNA sequences in this group, is very different from the tree distance in this group, right? And that is one of the pitfalls of using UPGMA, right? Because the tree distances don't match the actual observed distances. And like we spoke in length yesterday about, UPGMA assumes that every lineage is evolving at the same rate, and it gives you an ultrametric tree instead of a phylogram, right? The other thing about UPGMA is it's the only method that gives you a rooted tree, and every other method that we'll be talking about in this course will give you unrooted trees, which we will have to go and physically root, and we'll look at that when we do in the forthcoming days. So that is that with UPGMA. If anyone has questions thus far.
Yeah. So could you go to the previous slide, please?
Yeah.
Yeah, and could you explain how we got the tree in the orangutan and the rest of the human chimp and the gorilla clade?
Okay. So over here, if you look at where my cursor is pointing, we have already established using UPGMA that human and chimp are sisters, and sister to that is gorilla, right? So these three taxa form a clade, right?
Okay.
Whose common ancestor is here, right? Which is the root of a particular tree. Now we have to extend this tree to include orangutans.
Yes.
Correct.
Yes.
So now we calculate the distance between this clade, human chimp gorilla clade, and orangutans.
Yeah.
Right. How do we do that? By computing the distance, computing the actual distances between human orangs, chimps and orangs, gorilla and orang, adding them up and dividing that by 3. Right. So if you look at the previous observed distance, you get 7, 6, and 5, right? For these values. So human and orang is the distance is 7, chimp and orang the distance is 6, and so on. So it's 7 plus 6 plus 5 divided by 3, which is 6. Okay?
Yeah.
6 is the difference between this clade and orangs.
Okay.
And because we are using a method that basically uses arithmetic mean to assign branch lengths, 6 divided by 2 is 3. So each branch is 3 changes in length, right?
Okay.
Is that clear?
Yes.
Okay. Cool. Yeah, so I think we, any other questions on UPGMA, or I'm just going to move to neighbor joining. So the neighbor joining method is slightly more complex than UPGMA. So I'm not going to get into a lot of the equations that are shown here, but I'm just going to give you a gist of what is happening. Okay. So we use the same dissimilarity matrix that we got from our DNA sequences, right? The same as what we used for the UPGMA method. And this is our observed distance. And for neighbor joining, we calculate what is called the total distance for each taxa. Okay. For example, the total distance for humans is a summation of 2 plus 4 plus 7, right? The distance between humans and chimps, humans and gorillas, and humans and orangs. Okay. Similarly, the total distance for chimps is 2 plus 3 plus 6, which is 11. Total distance for gorilla is 12, and total distance for orangutans is 18, right? So here, if you notice, the reason we are picking total distance is so that we are not only looking at taxa pairs, we are looking at each taxa in the context of the other, right? So that's why total distance is calculated. So from total distance, we calculate what's called rate corrected distance. Okay. So this is how you calculate rate corrected distance. I'm not going to go into the equation here, but basically the rate corrected distance is a function of the actual distance between two taxa and the total distance of two taxa, right? So the rate corrected distance for humans and chimps, we calculate that to be minus 10. Okay, so why do we calculate rate corrected distance? So the reason we calculate rate corrected distance is actually to find the two closest taxa. So instead of using observed distance like we did in UPGMA to find the two closest taxa, which is human and chimp, we use a rate corrected distance, which basically corrects for changes or evolution across the group, and then we pick the two closest taxa. So in the dissimilarity matrix out here, can anyone unmute and tell me which two taxa are the closest after calculating rate corrected distance?
Humans and chimp?
Yeah, humans and chimps are the closest, and also gorillas and orang are equally close, right? Remember that these numbers are in the negative, so the larger the number, the lower the value, right? So humans and chimps are the closest, and gorillas and orangs are very close to each other too. So we can pick any of these two species pairs. We can either pick human and chimp to join them first, or we pick gorilla and orang, right? We go into the next slide. Let's say, assume we just pick gorilla and orang. Okay. Now we have the rate corrected distance using this matrix. and we have the total distance and the observed distance. So these are three different parameters that we have. To calculate the branch lengths that separate gorillas and orangs, we need to use the observed distance and the total distance, just like how we calculated rate corrected distance, right? So the branch length, so we presume that there is a hypothetical node X, which was a common ancestor to gorillas and orangs. And what we essentially are doing here is calculating how much evolution has occurred between X and G and X and O. And the amount of evolutionary change basically is your branch length, correct? So we calculate the branch length of XG, and then we calculate the branch length of XO. And using this equation, we find that XG is 1 and XO is 4, right? So if you kind of appreciate the difference between UPGMA and neighbor joining, even at this early stage, right? So between what UPGMA would have done is it would have figured out that the actual distance between gorilla and orang is 5, okay? 4 plus 1, 5. And UPGMA would have just divided that by 2 and assigned each branch a length of 2.5 each, right? However, with neighbor joining, it actually figures out how much evolution has happened across a branch. It does not have to be the same, right? So here in this particular instance, it actually tells you that gorillas have had just 1 change since the common ancestor, and orangs have had actually 4 changes since the hypothetical common ancestor, right? So even right here, you can gauge that neighbor joining does not use a molecular clock and kind of figures that every lineage is evolving at a different rate, okay? Now, moving on, now that we have this node X, we need to calculate the distance between X and the other 2 taxa that are in our groups, okay? So we recalculate, so the distance between, the actual distance between X and humans is basically this formula. We get 3 out of this, and the distance between X and chimpanzees is 2, right? So we calculate the distance between X and all the other taxa, and this is your actual distance, assuming that gorilla and orangs are in the same, like have been grouped together already, right? Now with this observed distance, we recalculate total distance like we did in step 1 in a couple of slides ago, and then we calculate the rate corrected distance the same way we calculated it a couple of slides ago, and we get this matrix. So this matrix here is a rate corrected matrix, okay? Unfortunately here all the distances are the same, which is -7. So basically you can pick whatever species pair you like, okay? So let us randomly pick species pair X and H. So we're trying to now join X and H using a branch, and we use a hypothetical node Y to be able to connect X and H. The reason for that being, X is a hypothetical node, and as you know, in a bifurcating tree or a dichotomy, every node can only lead to 2 taxa, right? Or every node can only distend 2 branches or stems. So we have to use another hypothetical ancestor Y over here and calculate distances between X and Y and H, right? So this is how we do that using this formula over here. I'm not going to go over it, but we figure out that the distance between X and Y is 1.5. The distance between Y and X is 1.5 again, right? So we get the tree that looks like this, where we have connected humans to gorillas and orangs, right? Moving on, we are left with one more taxa, which is basically chimps. So we have to connect chimps to that hypothetical node Y because it's free, right? So you add chimps to Y and you calculate the distance between Y and C as the branch length Y and C as 0.5. So the tree on your right shows you the completed neighbor joining tree for this group of 4 taxa, right? If you notice, branch lengths are completely different from each other, and they're not using a mean or an average of observed distance, right? The tree on the left, follow me here. The tree on the left is what we actually built using neighbor joining. Now, if you just represent the branches on this tree according to the distances, you'll get the tree on the right here. Okay. So if you notice, the branch from X to orang is really long because it's 4 changes, as opposed to the branch between Y and chimps, which is just 0.5. Right? And if you also notice, this tree is an unrooted tree, which means we'll have to root it. And if we choose to use the neighbor join, the longest branch or midpoint routing, we figure that the branch between orang and human is the longest branch, and the midpoint of this branch falls somewhere here, right? So we pull this branch across its axis, and we get a rooted tree that looks like this, right? So here, if you
Chaitanya,
yeah.
Can you just go back again, explain that midpoint? I did not understand of force the orang's midpoint.
Yeah. So with midpoint routing, what you generally do is, in an unrooted tree, you pick the longest branch, right? Or the longest path between 2 taxa. If you notice this unrooted tree, the longest path is from orang to human, right? This is the longest path or the longest branch, right? And in this path, you choose the midpoint. So if you calculate this path, it's actually 1.5 plus 1.5 plus 4. That is a total of 7. So this midpoint would be at 3.5. Okay.
Okay, got it now. Thank you.
And then you just pull this midpoint across its axis, and you get what is called, you basically get a tree that where branch lengths matter, right? Where branch lengths are actually accounted for. And if you see here, the distance between orang and the root is 3.5, right? Because you pulled it here at 3.5, because that's the midpoint. Yeah. Similarly, you see chimps, the branch leading to chimps is very short, and the branch leading to gorilla has 1 change in it from this node and so on, right? So this is a typical neighbor joining tree, which takes branch lengths and the amount of evolution that has occurred along every lineage, right? So moving on to the next slide, this is a comparison between UPGMA and neighbor joining, right? So we mainly included neighbor joining so that you guys can see the difference between the kind of trees that both methods generate and appreciate those differences. The UPGMA uses a molecular clock and assumes that every lineage is evolving at the same rate, and therefore every lineage is equidistant from the root, right? Neighbor joining, however, does not assume that, right? As you can see, chimps have evolved at a very slow pace from their most recent common ancestor from humans, whereas orangs have, so much of evolution happening along that lineage, right? So this is primarily the difference between these 2 methods, though both methods use distances to calculate to build trees. The other big difference, of course, is that UPGMA gives you a rooted tree, and neighbor joining gives you an unrooted tree, which you will have to root using the midpoint routing method, like we just did. Okay, any questions so far before we move to the practical session?
Chaitanya, I think this next slide explains it. Just the additive tree. Basically, the neighbor joining method gives an additive tree where the tree distance equals the observed distance, whereas UPGMA does not.
Yeah, that's another significant difference between UPGMA and neighbor joining. So the tree distance and the observed distance with the neighbor joining approach is the same, right? But with UPGMA, you get like the observed distance or the tree distance is completely different from the actual or the observed distance, right? So that's another big advantage of using neighbor joining as a distance method. So you guys can actually go and check out these equations in your free time. I didn't want to go over them because it gets pretty arduous to explain all these, complex. They're not really complex. They're very easy, but on the internet, it just becomes difficult to explain. But they're actually very intuitive to understand. So I urge you all to go and look at how these equations were built, and it's actually pretty straightforward. Okay. Any questions so far?
So I just wanted to add, do not use UPGMA method because it assumes molecular clock, and often you will get an erroneous tree. If you're going to use a distance-based tree building method, use neighbor joining.
Yep. Okay, I'm just going to take a 1-minute bathroom break. If you guys have any questions, you can ask Praveen and Roy, who's still on.
Yeah, guys, if you have a question, just unmute and ask.
Hi, sir. Sir, could you please explain the thing that you had said earlier about the observed distance and something like that? You don't know. Could you please explain
okay,
it?
so you see the slide, right? And there are two trees. The one on the left is UPGMA. The one on the right is neighbor joining. Okay. If you now calculate, say, the distance between human and chimp on the tree.
Okay.
That would be the tree distance between human and chimp. And if you add up the branches, 1.5 plus 0.5. So that is that gives you the tree distance, right? So similarly, you can calculate the tree distance between, say, gorilla and chimp. For example, between gorilla and chimp would be 1.1 plus 1.5 plus 0.5, which is 2. No, that is how much? That would be 3, right? The tree distance. Now, if you look at the observed distance between gorilla and chimp, the observed distance that you got from the sequence data, it is also 3.
Oh, okay. Oh, fine, sir.
So, yeah. So in case of neighbor joining, the observed distance and tree distance are the same for all species pairs. However, if you look at the UPGMA tree, the observed distance between chimp and gorilla is the tree distance between chimp and gorilla is 3.5, whereas the observed distance is 3. Right? So that is the problem with the UPGMA. The tree distance and observed distances do not match. They only match for human chimp, but for all other species pairs, they do not match.
Okay, sir. Thank you.
Okay, and yeah, that happens because it is assuming molecular clock, and it tries to fit the molecular clock, whereby it tries to make all the taxa equidistant from the root.
Okay, sir. Thank you.
So another thing that I wanted to add here is that both UPGMA and NJ just gives you one tree. So with UPGMA, of course, it is rooted already. NJ gives you an unrooted tree that you can either root using midpoint routing or you can use outgroup routing. But at the end of the day, it just gives you one tree topology, right? Whereas the other two tree building methods that are listed on the first slide, but we're not showing, those basically look at all other possible tree topologies. So, for example, here we're looking at a tree topology where human and chimp are sisters and gorilla and orang are sisters in the unrooted tree. But there is also a possibility, given these 4 taxa, there's also a possibility that human and orang are sisters and gorilla and chimp are sisters, or human and gorilla are sisters and chimp and orang are sisters. So these other tree building methods, like minimum evolution and least square, they look at all possible tree topologies. And Praveen was mentioning about additive tree, where you look at the tree distance and observed distance. So the least square, which is one of the tree building methods, the least square method actually looks at this particular character. It looks at the least discrepancy between your observed distance and your tree distance.
Okay, any other questions?
Okay, looks like everyone has understood everything, which is great. Okay, shall we move to the practicals? I'm presuming the answer is yes, so let's.
So Shruti is asking, but we can select outgroups prior in NJ. So once you get the unrooted tree, Shruti, once you get the unrooted tree, you can always root it based on your information. So in the taxa that you've selected, you know which the outgroup is or the outgroups are. So you can always select those as outgroup and root the tree. And we will actually do this exercise in the practicals.
Yeah, at
So
least
Emma,
for
yeah, Emma also has a question, but I'm not sure I understand it. Emma, can you explain what exactly you're asking? Well, I guess we. Oh, okay. Well, we'll try to get back to it later. Okay, I guess we move on then.
Okay, so we'll start with the practicals, and I urge you all to actually look at your screen rather than trying this out yourself right now. I know it's very tempting, but I'd rather you guys look at the screen and watch what we are doing here. And you can always try it at home after the session, because all of you have these software, all this data has been shared with you, and so on and so forth, right? Okay. So if you see this folder that I have, I'm going to start off with this software called Chromas. Okay, Chromas basically helps you clean up your chromatograms. So in the slides that we went through, you saw there was this step called direct sequencing, right? After your PCR amplification step, you send the amplified product and get it sequenced, right? So the sequences, when they come back to you, they'll come in what's called a file format called chromatogram file, like you see here, right? You see the file type.
So Chaitanya, we can't see your folders. So maybe you have to share entire screen.
Okay, hang on. I'll do that. Is it visible now?
Not yet. I think it might be some loading issues. Nothing yet.
Okay, I'm just going to share the screen then. I think there's some problem with the entire screen. No? No, Joy?
No.
Okay, I'll just share. Hang on. You see it now?
It's loading. Yeah.
Okay, cool. So I was saying that these files you see here, these are chromatogram files. You will see that the file type is chromatogram, right? Chromatograms are basically your sequences that have come in a sine wave format, and there is this program called Chromas that helps you edit chromatograms and clean them up and so on and so forth and interpret them. Okay, it's a very useful program. I've shared that tool with you too. You can install it and try this out later. So let me open one chromatogram and let's see how that looks. Okay. Can you guys see this?
Yeah, it's visible.
Okay. One sec. I just forgot one step. I'm going to close the screen for a brief moment. So I just want. So generally, what I would do is when I get these chromatograms from the company that does the sequencing or wherever, I would make copies of them. So even if I make a wrong edit, I have a copy. So I'm just going to do a Control-C of these two files and create copies, right? Because with a chromatogram, you will be editing and cleaning up your sequence. And in case you make the wrong edit and by mistake you save that file, your sequence is gone, right? There is no other copy of it. So you might as well have one copy in your one master copy in your desktop, and you will always have a copy in your email because those sequencing guys have sent you an email with the sequences in it, right? Always safer to have a copy is all I'm saying. So I'm going to open the copy and see how the sequence looks like. Okay, so if you guys can see the screen, you see a lot of garbled sine wave like things, right? So these sine waves, each sine wave represents one nucleotide, and you will see 4 of those in your sequence, right? So now we are at the beginning of the sequence that we have received from the sequencing company, and you see that the first nucleotide here is a C. Can you guys see this? It's a C, right? The second one is a G.
Hello, Chaitanya.
Yeah.
Can you hide that thing? Some notification is there.
Better?
Ah, done.
Okay. So you notice that the first sequence in your first nucleotide in your sequence is a C, and then you have a G, and then an A and a T, and so on and so forth, right? And this number over this number basically tells you the position of the sequence, right? So at position number 10 or site 10 in my sequence, I have a G, right? At site 20, I have a T, okay? At site 30, a G, and so on and so forth. Similarly, you see a little rectangle above the nucleotide, right? Above C, you see a little rectangle. Above G, here you see a little rectangle. And above A, you see that this rectangle is a little filled, right, with green paint. If you look here at T, this rectangle is completely filled, right? So what does this partial filling of rectangles mean? So basically, the sequencer is trying to tell you that for each site, this is how much it is confident about the presence of this nucleotide there, right? For example, the sequencer is telling you that, okay, at site 1 there is a C, okay, but I'm not at all confident about it. So that's why this rectangle is not filled, right? However, at site 28, it's telling you there is a T, and the sequence is telling you that I'm fairly confident that it's a T. Okay. Now, when you've got your sequences, you're going to be using these sequences to do all your analysis, right? To build your trees and so on. The important thing is you should not end up doing data analysis with unreliable data, right? So if you notice, maybe the first, like at least till the first 27 or 28 sites, there is a lot of garbage in the sine waves, right? The sine waves are all over the place, and sometimes you see, like multiple peaks at each site and so on, right? So you have to get rid of all this data because you cannot rely on this data for further analysis. Now, if you do not get rid of this data will be used in your tree building analysis, and it might just give you the wrong results, right? It might give you the wrong tree topology, right? So might as well do analysis with data that you're absolutely sure of, right? So it's like basically analyzing the number of new COVID cases, right? Which is a completely unrealistic and unreliable statistic, because the number of COVID cases is really an artifact of the kind of testing that each state or each country is doing. So you can't really go by that. So this is a little like that. So what if I just scroll along this sine wave, you see that a little later these peaks are kind of establishing themselves, right? And you also notice that these triangles are being completely filled, which means that the sequencer is absolutely confident that at site number 66, there is an A, or at site number 70, there is a G, right? That is what these, well-established tall peaks really mean, okay? So if you look at the rest of the sequence, you'll get a lot of nice looking towers. Looks a lot like Manhattan in New York. As you keep scrolling, you will get to the end of the sequence, okay? And at the end of the sequence, you again start noticing a lot of garbling here, right? For instance, at this site A, the sequencer is not very confident that at this site there is an A, because there could also be a T, because you see this other peak at this site, right? There is a red color peak which represents a T, okay? So there will be certain sites where there are multiple peaks and the sequencer is not really sure if it has given you the right nucleotide for that site, right? So as you go further down the sequence towards the end, you will see a lot of garbling that is happening, which is again completely unreliable data. So now what do? How do you make this data reliable? You snip off the unreliable parts from the leading edge and the finishing end, right? So you basically snip off those parts and you only retain the reliable part of the sequence. Now I'll just show you how that is done. So let's go back to the beginning of the sequence, right? And I'll say that, okay, I'll put my cursor at C, and I don't like this site because the sequencer is not confident and I don't see any comfortable peaks over here. So I'm just going to keep deleting these unreliable sites till I maybe get to this G here, right? This is a nice peak, and from here on, I see that the sequencer is fairly confident about the peaks and the nucleotides it's put in each position, right? So all these sites are really good, and I can use these sites to analyze data. So again, I go to the end of the sequence. Oh, there's a lot of garbage here and all that. So we're just going to ignore all this. It's not there. So I go to the end of the sequence, and I again see that there's a lot of noise happening here. So I again start deleting these sites. Yeah, not really happy with any of these because even the confidence of the sequencer is pretty low. Till I maybe get to site number 520, where I have a nice tall peak, and the sequencer is also fairly sure about the presence of a G there, right? So now I've deleted unreliable data from the beginning and from the end, and I've just preserved the reliable data from in between. Everyone with me so far? If anyone has a question, you can ask right now.
Chetan here, Akshobh here.
Yeah.
My question was, if you see there are unreliable data in between as well, so should we keep those or we have to delete those as well?
Sorry, say that again.
Just noticing that there are unreliable data like in between the snippets. So should we?
Yeah, so the way to interpret this is as long as there are distinct peaks, right? You see these distinct peaks out here, right?
Mm-hmm.
Then the sequencer is fairly confident about the presence of that nucleotide at that site, right? So what the nucleotide is saying here, for example, you consider this site G, which is site number 212. It's saying that it's very, very confident there is a G, okay?
Mm-hmm.
Maybe 95%. But you will also see a little bump here, which represents A, right?
Mm-hmm.
So it's saying that 95% it's a G, maybe 5% it's an A, and there is very, very little chance that this is either a C or a T, right? So that's why you see this little garbling in the bottom, right? So as long as you see these distinctly clear peaks, you can be fairly sure that the nucleotide is, the sequencer is good with all these sites, right?
So if you just look at 259.
259. Okay, this one, yeah.
So that's like it's not sure that if it's a G or not. So
yes.
So would we delete that as well, or just delete the starts and ends?
only at the beginning and the end do you delete, right?
Okay.
Never delete in between.
Okay.
Right? If you delete in between, it changes the kind of, it basically changes the sequence itself, right? And that is not good, right?
Okay. Okay.
Yeah.
Chaitanya, if I can add,
Sure.
so that particular site has 2 peaks, right? And you are not able to figure out the program has called it G because the G peak is slightly higher than the green peak. But in situations like this, you should not just assume that it's G. What we normally do is for every marker we use, say when we do PCR amplification, you use 2 primers, right, to amplify a particular marker. So it is a good idea.
Sequence too.
I'm sorry?
I'm going to show them the reverse sequence and how we do the reconciling.
Okay. Fine. Okay, fine. So basically, I was trying to say that we sequence both forward and reverse. Okay, fine.
Chaitanya, can I ask a question? Yeah.
Sure, go for it.
Yeah, at 280, there is like proportion between all this A or T is very less. So how it will interfere with my whole data set, what I am selecting?
So for the moment, we retain this data, right? Because we don't delete in between. And you see around this T, there are a lot of nice peaks. Okay.
Yeah.
But for cases like the one you brought up, like, is this a T or is this a G, right?
Yeah.
The G is like almost halfway through here. And similarly, in site 259, the sequencer is very unsure about the G, right? It could either be a G or an A, right? For such situations, we have another mechanism of fixing this, right? So we will get there. We will get there when we get there.
Sure.
Okay?
Chaitanya,
you may ask a question.
You just understand that. One second. So for the moment, just understand that the sites in between should not be touched or should not be deleted, right?
Hello.
Because if you delete a couple of sites in between, then you are basically meddling very badly with the data, and you don't have a reliable sequence anymore. Okay. So what was your question? Sorry.
Yeah. Can we take multiple short sequences, say for example 20 to 259, one sequence, then 270 to 380, like that?
Sure. You could, but you will only have that many characters to analyze. So if you take 270 to 300, for example, you only have 30 characters to look at.
Hi Chaitanya.
Yeah.
I think Shoibal sir wanted to ask you, like, and then this 30, and then again you go back and take some more and add. That is not possible, I think, because you are missing. Now my question is, like, you are going forward with this one, and then you align in Mega with reverse primer.
So we'll come to that. We'll come to the reverse bit. I'm going to show you the reverse bit.
Okay.
You're not done with this. Yeah.
Okay.
So bottom line is, don't delete from the middle. If you delete from the middle, your data becomes unreliable, right? And it won't align with the rest of the species in your group that you're trying to build a tree with, right? Now, once you've cleaned up cleaned up the leading ends and the trailing ends, you save this chromatogram, right? You save it, and it saves as a chromatogram. And then you also export it into a format called FASTA. So when you click on export in the Chromas program, it automatically, by default, tries to save it as type FASTA, right? So now we'll say, yes, let's save this as FASTA, because FASTA is the file format that programs later in the pipeline like Mega and so on recognize. Mega and other programs, they don't recognize chromatograms at all. So they need formats like FASTA. So let's save the sequence as FASTA. Okay. Now, like Praveen was mentioning, and a lot of you had raised that question, right? So what do we do with some of those some of those sites in between in your sequence that the sequencer is not really comfortable with, or the sequencer is not really sure of, right? So just for that, what we do is we generally use two primers for every marker, right? One is the forward primer and the other is the reverse primer. Okay. Now the forward primer will give you the forward sequence of the gene, and the reverse primer will basically give you the reverse sequence. Okay, I can just show that quickly in a notepad of how that works. Like, I'll just take a quick example so that you guys understand this. So let's say I hope all of you can see my notepad, right? Okay, if I can improve.
Yeah,
okay.
It is.
Okay, so let's say my forward sequence looks like this: ATT, GCT, AAA. Okay, let's say hypothetically my forward sequence looks like this, right? The reverse sequence that corresponds to these sites in that gene, right, will look like this, because as you know, the molecule A binds with T, right, and T with A. So there will be a T over here, there will be an A here, there will be another A here. G binds with C, so it will be a C over here and a G here, A, T, T, T. Are you guys with me so far? If you want me to go over this again, I can do it again.
People are typing yes, so it's fine.
Okay, great. So
once more, Jigme says once more.
Okay, so let's say your forward sequence looks like this. Okay, let me just tap this so it's in the center. So let's say your forward sequence looks like this. Okay, so ATT, GCT, AA. Now, when we get the reverse sequence, right, it will look something like this. So A binds with a T in your DNA, right? In your DNA structures. So it will be a T over here, and T binds with an A, so it's A here. G binds with a C, so it will be C. C binds with a G, it will be a G, and A followed by T, T, T. Right? Because DNA is two-stranded, right? And each strand is basically a sequence of nucleotides. And if in one strand you have A at a particular site, in the corresponding site in the other strand, you will have a T. Right? So basically, that's what your reverse sequence gives you. Okay? I hope I'm
okay.
Okay.
Yep.
Now, we have this problem. Now we have this forward sequence, right? We have ATT, GCT, AA, ATT, GCT, AA, but we are not very sure of some of these sites because the sequencer has given us very low confidence for, let's say, these three nucleotides, right? So that is the reason we do a reverse sequence. And what we're going to do is basically we take the reverse sequence and do what is called a reverse complement of the reverse sequence, right? So what does reverse of reverse do? Takes you forward, right? So if you do a reverse complement of this reverse sequence, you will see the same as the forward sequence, ATT, GCT, AAA, right? So this is what the reverse complement does. And then we get rid of this reverse sequence and we only use the reverse complement and match these two sequences, right? We just match these two sequences to see if every site has the same nucleotide. And in case one of the nucleotides with very one of the sites with very low confidence here was, let's say, an A, right? And the reverse tells you it's a C, then we can debug this and we can decide which is the right site by going back and looking at the chromatograms. Okay, so this is the idea. All of you with me so far?
Yeah, so along with that, I have a question. So what if our gene have an internal primer along with that? How we align that also? In addition to the forward and reverse primer, if our gene sequence have an internal primer, how do we tackle with that problem?
I'm not really sure. So Praveen or Roy, can you take that question? Praveen, Roy? Okay, I think there's some.
Oh, wait, sorry. I just realized I was on mute. Yeah, so you're saying use an internal primer because you have a really large gene that you want to sequence, right? So you will still have the forward and reverse. Even if you have the internal primer, you'll still have the forward primer and the reverse primer, right?
Yeah.
So you still get two different sequences, right? And you
Yeah.
can still do the same thing that Chaitanya has said. It's
Okay.
just that with an internal primer, maybe you'll have two forwards and one reverse, but you'll still at the end of the day you will have two sequences to compare with.
So should we consider the internal primer while aligning or doing that?
So we are not considering the primer sequence. You're just basically you're looking at the sequence that that has come from the sequencing, from the Sanger sequencer. That's it. Your primer, the only work of your primer is getting the sequences. That's it.
So again, you should remember that if you're using forward and reverse primers, regular forward and reverse primers, and let's say some of the samples that you've used an internal primer, right? Now that's going to be useless, right? You can't align those sequences properly. I mean, you can align them, but you'll be losing a lot of data.
Sir, actually, the internal primers are used when the sequence that we have to get is more than 1,000 or something base pairs. So we use internal primers in between to get a more consistent, reliable kind of, sequences. And so we should align the internal primers with the forward as well as the reverse, no, sir? Then only you will get the aligned sequences, no, sir?
Correct. So with Sanger sequencing, generally the way it works is you will get a lot of noise after, say, 750 base pairs, 750 nucleotides. So which is why a lot of people, when they face problem in getting like a large chunk, they use internal primers. But just remember, even if you use internal primers, you're still using a forward and a reverse, right? So let's say you're breaking 1,000 into 500 and 500, right? So you still have a forward and a reverse for the first 500. You still have a forward and reverse for the next 500.
Oh, so first we should align the first 500 reverse forward and reverse, and then.
Yeah, so you have to do those separately.
Oh, okay.
You just think of them as two different data sets altogether.
Oh, okay, fine.
Hi, Chaitanya, Aniruddha, both. I have a question, Jayaditya here. So, if in forward also for the third nucleotide, for example, the T in both the cases, in both the chromatogram, if the confidence is very low, in both in case of forward and reverse, and we cannot let go the sequence, or maybe sometime we have these missing gaps there.
Yeah.
So is it advisable that I take 6 more sequences of similar or same or similar species from NCBI and then align them, and if they are like every other sequence for the next 5 sequence, all those data are T, can I be confident that this should be T only and keep it like that?
Absolutely not. Absolutely not, because you're basically making an assumption, and assumption is basically like, not a great thing to do, right? So you can't assume that there is a T there, but there is another way of kind of. So basically, what you need to do is you need to ignore that site, right, from your analysis. If both your forward and reverse give you very low confidence for that site for a particular nucleotide, then you need to kind of ignore that site, and there is a way of specifying that in a sequence, and we'll get there when we deal with Mega.
Yeah, so you can either treat it, I think Chaitanya will talk about it, you can either treat it as missing because you're not confident. You can either treat it as missing data, which will be dash, or you can treat it as N, which can be either A, T, G, or C.
Okay, thank you.
Okay, and then there are also other cases which are you going to talk about degenerate nucleotides, Chaitanya?
No, I don't think so.
Okay. Okay, so so in certain cases you might have a case where, for example, so right now they're exactly similar, the forward and the reverse complement of the reverse, but say for one of the nucleotides, for one of the sites where there's a ATGC in the forward and there's an ATTG, let's say there's a T in the reverse. So you have, and you check both the forward chromatogram and the reverse chromatogram, and it looks like the peak of C and T is equal.
Okay.
Right? So in cases like that.
Let's quickly do that as an exercise and finish that off.
Sorry?
We'll just quickly do that exercise.
Yeah.
That would be great, yeah.
Yeah, okay. So I'm just going to open the reverse chromatogram now and clean it up quickly in the interest of time. I don't like these sites because they don't look great. So maybe from somewhere here I could start my sequence and I go right up to the end. All this is nonsense data. So I'm just going to clean all this up. And even these, if you see, these peaks are not great, so I'm not going to keep them. Okay, from here on it looks okay, right? So I'll just like we did with the forward, I will save this chromatogram and I will also export it as a FASTA file. Yeah. Now we are done with the chromatograms, right? Now we need to see if our data is actually consistent, right? So I'm going to open this program called Mega. Okay. So Mega is a software that we all primarily use to, check sequences and do our alignments in, right? Mega is a very useful program to have, but it's kind of not useful for anything else. We'll just shortly talk about that. So what I'm going to do now is I'm going to say, like edit build alignment and say create a new alignment. Okay. Are you building a DNA or protein sequence alignment? We're building a DNA alignment. And it'll give you a window like this. So from here, I go to edit and I go to insert sequence from file. Okay. So then I choose my forward sequence first and I open it. Now, like Roy spoke about, we have to compare this forward with the reverse complement of the reverse sequence. So I have to also load the reverse sequence. So let me go back and insert the reverse sequence also, right? So now I have the forward sequence and the reverse sequence, okay? But I have to do a reverse complement of the reverse, right? So I choose the reverse sequence by just clicking on it and do a right click. It will give me the option to reverse complement over here, right? So I do a reverse complement. And now I have the reverse complement of the reverse sequence, which should look exactly like the forward sequence. And once I'm done with this, I have to now align the two sequences, just so that site 1 in the forward sequence is actually site 1 in the reverse sequence as well, right? And site 243 in forward is site 243 in reverse. So basically alignment ensures that you're comparing apples with apples and not apples with pineapples, right? So I click on this alignment tab and say align by cluster. So it throws out saying nothing selected for alignment. Should I select all? And I say yes, select all. Right? So now it's performing the alignment and it is aligned my forward sequence along with my reverse complement of my reverse sequence, right? So this is what that looks like. If you notice, it has left a huge gap in the forward sequence, right? And then it starts aligning with this side, right? That is because of the way we cut the sequences on either side in the chromatogram, right? So that's the reason for this gap, and you don't have to panic if you see this gap, right? Now, if you look at mega, so each little rectangle here represents a site in your sequence, right? And in our use case here, every site should have the same nucleotide. For example, this is site 52. If you look at the bottom left of your screen, it tells you the site number, right? There. So this is site 52, and site 52 has G in both sequences, which is great. Similarly, 53 has a G, 54 has C, and so on, right? Now, if both sequences have the same site, have the same nucleotide in that site, mega puts a star on top here, as you can see, right? So it basically helps you out so that you don't have to go and check every sequence and in every site. You just have to look out for the stars to see if all sequences have all sites have same nucleotides in that site, right? So in our case, we are trying to match forward and reverse to see if there are any discrepancies. So ideally, all sites should have a star, correct? Basically, all sites should have the same nucleotide. So let's scroll across the sequence and see if all sites have the same nucleotide. So far, so good. All sites seem to have the same nucleotide. However, if you look at this site, right, the forward sequence has an A and the reverse sequence has a G. The reverse complement of the reverse, of course, has a G, right? This also should have been A, and then this would have been a star. So here at this site, which is basically site number 295, we have a discrepancy, right? Now, how do you fix this discrepancy, right? You have to go back to the chromatogram and see what that tells you, okay? So let's first go to the forward sequence chromatogram, right? So remember that this is site number 295, and your gaps in the first sequence end at site number 51, okay? So to be able to get to the right site in the chromatogram, right, I have to calculate the site, calculate the right site in the following way. So the site where the discrepancy is site number 295, right? And I have 51 gaps, so I subtract 51. So 244. So site number 244 is what I should be looking at in my chromatogram to see which nucleotide is present there. Okay. So let's do that. Let me open the forward chromatogram.
Hey, Chaitanya.
Yeah.
There is an easier way to do this. So in the chromatogram, in the chromas file.
Yeah.
So in the reverse complement. If you open the reverse complement and you just, if you open the reverse and you just do a reverse complement.
Okay.
Then you'll have what you have on mega, right? And if you copy and paste a bunch of sequences where, for example, there was either an A or C, right? Difference. So just starting from A till about another 5 nucleotides, just copy, and there is a find option on the chromas file, right?
Roy, I think I can learn this method after this session. I'll just show them the way I know it, so that
okay.
no used. So we were at site number 244, if you remember. So I picked the forward sequence of the the chromatogram of the forward sequence, and basically just go to site number 244 and see what's there, right? So this is site number 240, 41, 42, 43, and 44. Okay. So here, site number 244, if you see it is A with very low confidence, right? And you see that there are 2 peaks, and the sequence is basically saying this could either be A or it could be G, right? So even with the chromatogram, we are not sure what the site actually is. Yeah. So in such a scenario, we need to go to the reverse sequence and see what is there in the reverse sequence, right? So let me open the reverse sequence in the chromatogram. But now we'll have to look at the sequence from behind, right? So this is site number 295. So if you open your reverse chromatogram, oh shit. So if you open your reverse chromatogram, you go to the end, you see there are 540 sites, right? So you basically subtract 295 from 540, right? You get 245, and to that you add 1 because you're going from behind, right? So you have to be looking at site number 246 in your reverse chromatogram. I hope you all of you are with me so far, right? Basically, we are going backwards here, right? So I go to site 240. That's 241, 242, 43, 44, 45, right? So here you notice that it's very confidently a G. Oh, I'm sorry, it's this site actually, right? So here, it was 246, right? Yeah. So at 246, you have a C, right? And very confidently so. So you can trust the reverse sequence and say that, okay, at site 246, we have a C. And what's the reverse complement of a C? It's a G, right? So now we know that this site should actually be a G and not an A, right? So we can actually go and modify your forward sequence, convert it to a G, and then you're sorted, right? Everyone with me so far? So this is how you basically clean up sites that are contentious between forward and reverse. Yeah. So for every marker that you choose to sequence, it's prudent to use both forward and reverse sequences just so that you're able to clean up data in this way, right? And if there are any more sites in the sequence that kind of are contentious, then you go for it. Then you have to basically repeat the same process and hope that there are not too many sites that are contentious. Okay, I hope all of you were with me so far.
Chaitanya, at the end, if you go there, you will see a missing data. Should we change it with the one which is at the top, T?
Not necessarily. You can just leave that be.
Okay.
So if there is, like, let's say there are a few missing data in your sequences, the analysis will ignore that missing data, right? It will basically ignore that site. So you don't have to really worry about this too much. Yeah. And
okay, so Roosbay has a question. Any likelihood that both forward and reverse would be unclear for a base?
Well, there is a great chance, right? There's a great chance that forward and reverse are unclear. So let's just pick a random site here, which is site number 331. Let's say my forward and reverse chromatograms are fouled up and nothing is clear. So like Roy was mentioning earlier, right? I can't use this site because the site is unreliable. So I would basically put what is called an N here, right? On both forward and reverse. So what N denotes is basically it's telling the programs that are going to use these sequences in the future that there is definitely a nucleotide at this site, but we don't know which nucleotide. So the N basically denotes the presence of a nucleotide. And a lot of these tree building algorithms would kind of ignore these sites with N in them. Okay.
So N is also for like any base. Yeah, so N is for any base, but you can also use other IUPAC, degenerate nucleotides. For example, if there's a similar peak for A or G, right? So there is a particular letter for that, R. So you can just put R there. So that R means that it can't be C, it can't be T. It has to be either an A or a G.
Yeah, Praveen, you wanted to say something?
No, nothing. I mean, basically that N means it can be A or T or C or G. That's all.
Yeah.
I mean, I think that's about.
Yeah. Okay, so that's that with cleaning up your sequences and dealing with contentious sites. Let's quickly move to what Mega can do. Okay, so what I'm going to do is I'm going to show you a sequence alignment. Yeah.
Chaitanya, just now Roy said AG is equal to, if I am not sure about A or G, it's R. And T and C, what would be?
Oh, there are IUPAC codes for every combination, right?
Just
Okay.
Google degenerate nucleotides or IUPAC nucleotides. You'll get the.
Okay.
Yeah.
Okay.
Cool. So I'm just going to load an alignment that we have already built. I'll close this. I don't want to save this anymore. Okay. So now I'm going to open a saved alignment session. Okay. So in my practicals folder, you open the Mega folder. You'll see a primate CT-B file with extension MAS. Okay. So that's the file you're looking for. That's the Mega alignment file. I'm going to choose that and open it. And you see, this is what an alignment in Mega looks like, right? On the left, you have your like terminal nodes or your taxa, and the corresponding alignments are over here, right, on the main section of the screen. Like I told you, if all the taxa have the same nucleotide for a site, Mega puts a star on top, right? So for the first 5 sites, you realize that all these taxa have the same nucleotides for the first 5 sites. Of course, the first three sites represent what's called a start codon. I hope you guys know what a start codon is and what the stop codons are. So the gene, basically the gene would start with a start codon, which is represented by these 3 nucleotides A, T, and G, right? So, by the way, I forgot to mention that the alignment we're using is from cytochrome B, and cytochrome B is a protein coding gene, okay, which means that nucleotides are divided into sets of 3, and each set is called a codon. Okay, for example, this is the first. Watch what I select. This is the first codon in this sequence, right? And incidentally, it's also the stop codon. But it does not always have to be the stop codon. You can start with any codon. But this is the first codon. This is the second codon, the next 3. And the next 3 will be the third codon, and so on. Similarly, you'd say that site number 1 is codon position 1 in codon number 1, right? Site number 2 is codon position 2 in codon number 1. And site number 3 is codon position 3 in codon number 1, and so on and so forth. And each codon basically codes for an amino acid. Okay. Now, this is how your alignment will look like. As a first step, just to see, just to do like a sanity check of this alignment, let's translate these nucleotide sequence into amino acids by clicking this translated protein sequences tab, right? So when I click that tab, it asks me a stupid question to which I replied no. And then it asks me a more pertinent question. So it says, is the current genetic code standard? Is this correct? So by standard, Mega generally means that is this a nuclear gene, right? But this is not a nuclear gene. We know that this is cytochrome B and this is a mitochondrial gene. So I say no, and Mega gives me the option of choosing various categories of genetic material, right? So this we know it belongs to a primate, so it's a vertebrate and it's a mitochondrial gene. So we select vertebrate mitochondrial, okay, and we click on OK. Now it has translated my nucleotides into amino acid sequences, right? Now, if your amino acid sequences look very different from each other, right? Then you have a problem with your DNA sequences, right? One way to do a sanity check is to basically see if all the amino acids look pretty much the same. Of course, there will be the odd difference, like in this site that I'm selecting. You see that instead of T, Papio hamadryas has M, right? But that's okay. The one-off change is fine and it is expected, but there can't be too many differences in your amino acid alignment. So if there are too many differences in your amino acid alignment, that means, you know, some goof up has happened somewhere and you probably are better off by resequencing all your data, right? So this is one way to sanity sequences. And then there is this other little that could happen with a protein coding gene, right? Here in our sequences, we know that we are starting at codon position 1, right? And this is the start codon, okay? Sometimes when, let's say, you've deleted a lot of inappropriate data from your chromatogram, right? So you've deleted a lot of leading nucleotides and ending nucleotides. Your sequence, and then you align your sequence, your alignment might actually start at the second codon, right? Or in certain cases, even from the third codon. But
Chaitanya's second position of the codon.
I'm sorry. Second position of the codon, or it might start from the third position of the codon. Now, when that happens, what is actually happening is the reading frame of your sequences has changed, right? And once that changes, you will see a lot of garbage or garbled amino acid sequences when you actually translate. And you'll see also what's called premature stop codons in the middle of a sequence. Okay, stop codons ideally should be at the end of your sequences. You can't have stop codons in the middle. Okay, so mega, it represents stop codons as stars. So they are easy to identify when you translate to amino acids. So now, when we are at the right reading frame, we are reading from codon position 1 and we translate, right? We see that there are no stop codons. If there are stop codons in between, mega would put a star there. If you look at these sequences, there are no stars anywhere in the middle of these sequences. It's only amino acids. So we are pretty much all good here, right? Let me show you what happens when we change the reading frame. Okay. So the correct way to change the reading frame is to convert it to the MEG format and all of that. But since we are running a little short of time, I'm just going to be a monkey and delete the first site. Okay. Now your reading frame starts from the second codon position, right? Now when you translate to amino acid, see what happens. You see all these stars in between, right? And all these stars have suddenly appeared in the middle of these sequences. So the reason for this is because the reading frame of the sequence has changed, right? And all the stop codons, which are basically, I think, TAG, TGA, and TAA are occurring here. So mega is interpreting them as stars. So this is not a good situation, right? So when you get a situation like this, you have to change the reading frame to be able to interpret your sequences and basically get rid of these premature stop codons in your sequences, right? Is everyone clear, or do you have any questions?
Chaitanya, Jayaditya here.
Yeah, Jayaditya.
So for some instance, I have actually in practical, for example, I have this situation where my sequence actually starts from T, that your sequence as of now, and I get premature stop codons. So is it ideal that I try of deleting, because the only option would be there is something wrong with the reading frame. There may be something. So is it ideal that first I try to delete the first
no, actually you shouldn't delete. So I was just being lazy and I deleted that first site. So maybe I should just show how it is actually done, right? So okay. So what I'm going to do is I'm going to open another file. So I can save this in a format called MEG format, right? So I go to data and I export my alignment and click on mega format. So by default, it saves it as a .meg file, okay? And I'm going to put it in the practicals folder. So input could be anything, it doesn't really matter. Encoding nucleotide data. Yes, so I have saved it as .meg. Okay, so now let me close this alignment here. And then from Mega, I go to edit, build alignment, create a new alignment, DNA. From data, I open. So let's open the MEG file. Okay, I'll just open any MEG file. I don't know where I saved the other one. Oops. Okay, so I'm just opening this MEG file. Okay.
Chaitanya, I think it's better just to directly click on the MEG file, because now it's again open mass.
Yeah, actually, if I okay, cool. So let me just open the MEG file. This is the one we created right now. Okay. So I can look at the data by clicking on this tab, right? And here, if you look at the MEG format and the way Mega interprets this, it's saying that TGA is the first codon in my alignment. CTA is the second, CCC is the third, and so on and so forth, right? Now, I can actually change the reading frame using Mega without deleting the, without deleting a site to be able to, correct for.
Chaitanya, sorry, one more thing, yeah? Can we see all the sequences, because all those dots might be confusing? So just click on what is it? I forget the no.
Yeah.
I think yeah. That's better.
Yeah. So and you can go to data and select and edit gene. Select and edit. I think it's select and edit gene domains. Okay. Yeah. So here, if you look at this screen, right, it says there is one gene or domain, and it starts from site number 1 to site number 1139. And there is also this column which says codon start is first site, right? But we know that this is giving us premature stop codons. So let's move the reading frame from site number 1 to site number 2, right? So here I would move it to the second site, right? And close. Okay, and now when I translate, there should be no stop codons. But I see that there are still stop codons. You see these stars here. So still my reading frame is incorrect. I'm not reading from the right codon position. So I go back. What did I do?
Select code table.
Oh, okay. Okay, so I go to edit taxa. Oh man, sorry. I go to select edit gene domains and so on, and again choose the codon site and move it to third site, and hope that this really is the real start of a codon, right? And I've basically moved my reading frame to the third site, which is this site, right? This site. Now when I translate to amino acid, oops, it was amino acid already. Okay, so now I translated that to amino acid, and I see that.
Chaitanya, just change the code table. It might not be vertebrate mitochondrial.
Oh, this is SIDB for primate.
Yeah, so it might be on standard.
Okay. It is vertebrate mitochondrial.
Oh,
it retains the mass. When you save the mass file into mega, it retains it, I guess.
okay.
Okay, anyway. So now we've translated into amino acid, and you look at the sequence, it's all clean, right? There are no stop codons anywhere. So now you know that you're reading the sequence right, and you've partitioned the sequence correctly into the right codons. Okay, so Jayaditya, does that answer your question on how you?
Absolutely.
Okay, great. So you don't really have to delete the leading sites to get to the right reading frame. You can actually retain those sites and just change the reading frame to the right codon position.
Okay,
yeah.
Thank you.
Cool. Now let's.
Chaitanya.
Yeah.
This is Shubham here.
Yeah, Shubham.
What if the reading frame of just one of the 11 sequences is it's not correct? What do you do for that?
It will never happen in such a way, right? That is because you're already aligning the sequences.
Okay, I got it.
So site number 241 in sequence Aluta Belzebub or whatever will be the same. Basically site number 241 will be aligned across all these taxa in your alignment. That's why it's called an alignment in the first place, right? So by essence, you're not comparing different sites across different taxa. You're comparing the same sites.
Okay, yeah, got it.
Okay. Now let's quickly look at how we can build some basic trees using Mega. So I close this and I go to this Mega window and click on. Let's first try something called compute pairwise distance. So basically, this will tell you the number of differences between each taxa in your data set.
Chaitanya, I'm sorry, can I just interrupt for just 1 minute?
Sure.
So I just wanted to point out that all this analysis that Chaitanya just showed is for protein coding genes. If you are working with non-coding markers, then you don't have to do all this analysis, right? And the other important point is this particular marker that Chaitanya showed us is mitochondrial. So mitochondrial genes don't have introns. And if you're using nuclear genes, you have to make sure the introns are also properly circumscribed in the or the program knows where the introns are. Otherwise, again, you might get a protein sequence that is messed up because introns don't code for a protein, right? So there are other complications. We have just shown you the most simplest sort of a situation. So if you are so two take-home messages. One, if you are using non-coding regions, basically regions that don't code for anything. For example, the D-loop region of the mitochondrial genome, then you don't have to do all this. It's only for protein coding genes. And in case of protein coding genes, if you're using nuclear genes, you have to specify the introns because only the exons code for amino acids. Thanks.
Okay, yeah, thanks for that, Praveen. Yeah, so that's very important to be able to know which marker you're using. And only if you're using a protein coding marker, you need to do all this gymnastics. If you're using regions where, like Praveen said, where non-coding regions, then you'll have to specify to Mega that starting from this site, let's say site number 35 to site number 86, it's a non-coding region or something like that. You have to specify that in Mega, and Mega allows you to do that. And Mega also allows you to remove non-coding regions from the analysis, right? It basically won't analyze your non-coding regions unless you ask it to. Okay. So moving on. Yeah.
How do I understand a non-coding region in that?
You will have to know the layout of the gene.
Okay.
So generally, if you download sequences from NCBI, that file, that FASTA file, will have information about where the coding regions are and where the non-coding regions are, and you can just specify that in Mega. And if you're using the same primers to sequence your taxa, you can use that information, right?
Okay.
Okay, now I'm going to do a compute of pairwise distances. Don't really worry about all these options it's given here. Roy will talk about some of these tomorrow. What we're going to use is only something called p distance. Roy will talk about p distance tomorrow, so we'll not worry about that. Let's just quickly compute pairwise distances. There you go. It's computed pairwise distance and it's given you a matrix. Okay. So basically, what this says is the distance between Aluta and Calithrix is 0.178. Okay, and distance between Aluta and Gorilla is 0.22. Okay, and so on. So basically, you can kind of you should make the inference that Aluta and Gorilla are 22% divergent for the cytochrome B gene. Okay, so that is basically how you infer this distance matrix. Okay, but here it is shown in the format of p distance, so that's why it's a decimal number. Yeah. Now there are a few other gymnastics that you can do with Mega. So let's quickly try and build a UPGMA tree, a tree that we are familiar with, right? So it asks you for a lot of options. Ignore those options for now and compute. Just click on compute. And there you have your Mega computed UPGMA tree, right? Notice that how the tree is already rooted. You don't have to bother. And you know how the tree is ultrametric and so on and so forth. Okay. You can also construct neighbor joining trees. Let's just try that because we used neighbor joining today. Let's compute and try and get a neighbor joining tree. So here, this is your neighbor joining tree. Now, if you notice, this tree is not rooted, right? There is no semblance of a common ancestor anywhere. So we have to physically root this tree. So how would I do that? I know that
Chaitanya, can I just interrupt?
Yeah.
This tree is rooted because unless you specify the root, it uses midpoint.
Oh yeah, right.
Yeah, and you know, these bootstrap supports are there, no? So maybe we should just build it again without the bootstrap support. But anyway, it doesn't matter.
Why does bootstrap support matter here?
No, what has happened is when you ask for neighbor joining, the bootstrap icon was also activated. I think.
Yeah.
That's why. But it does not matter anyway.
Okay. So like Praveen says, this is actually rooted using the midpoint routing, which neighbor joining does for you. But if you want to actually do outgroup routing, that is possible in Mega. So you select the branch leading to the outgroup, which is this branch, right? We know that this is the outgroup because all these are, I presume, old world monkeys. So we choose the branch that leads to the old world monkeys group.
No, wait, Chaitanya, wait. I don't think
it's Loris and Lemur.
It's Loris and Lemur, yeah, these two.
Okay, sorry, I don't know this group very well. So that's the importance of taxonomy, right? So if you don't know which your outgroup is, you'll be doing all sorts of things. So Lemur and Lorises are the actual outgroup, I'm told. So I will be rooting this tree using this branch. All with me so far. And in the left, Mega gives you the option of, there's something called place root on branch. So I've selected the branch and I'm going to click on place root on branch. So now this tree is rooted using the outgroup method. We have a bunch of apes and some old world monkeys and I think new world monkeys, and then lemurs and lorises as outgroups. Is that clear, everyone, so far? Any questions? Don't worry about the numbers you see along each of these branches. We will talk about them tomorrow.
Yeah. Okay, I don't see any questions on the chat box.
Okay, so I guess we have overshot by a mile today. So if anyone has, I think I'm going to stop right here. Did I miss anything with Mega, Roy, and Praveen?
No, I think what was supposed to be covered today is pretty much covered. I just wanted to add that routing is extremely important, and all the programs would spit out a tree. You have to make sure that it has been routed properly. So what Mega did here was it did midpoint routing when it first gave you the tree. Now, when you look at it, you realize, oh, wait a minute, the outgroup is Loris and lemurs, the prosimians. These are the most so-called